Welcome to soft mystic stories somewhere deep in the Amazon basin. Underneath centuries of sediment and forest floor, researchers pulled fragments of human bone out of the earth. Bones that were 7,000 years old, maybe older. Bones from people who had lived and died in one of the most isolated interiors on the planet, thousands of miles from any ocean, thousands of miles from any other known civilization at the time. Routine work, in a sense. Archaeologists find ancient remains. They document, they analyze, they publish. That's the job. Except when they extracted the DNA from those bones and ran the comparisons, something came back that had no business being there. A genetic signal, a match, not to populations from North America, not to groups migrating down through Central America, the way every textbook tells you the Americas were populated. The match pointed across the Pacific Ocean to populations living on the other side of the world. That's the kind of result that makes scientists check their equipment twice. Here's why it matters. DNA doesn't lie, but it does require interpretation. And interpretation is where things get complicated and fascinating in equal measure. When geneticists say two populations share a signal, they don't necessarily mean those people sailed from one continent to the other and shook hands on a beach. They mean something connected these populations at some point in deep time. Something left a biological fingerprint that survived thousands of years and showed up in a lab in the 21st century. What that something was, when it happened, how it happened, and whether it happened once or many times, those are the questions that keep researchers arguing, revising, and rebuilding their models of human prehistory from the ground up. Because migration stories aren't just academic puzzles, they're the architecture of human identity. When you rewrite them, you don't just move dots around on a map, you change everything downstream. You change how we understand the peopling of entire continents, the complexity of ancient seafaring, the timeline of when humans were capable of what. A single DNA match in a single set of ancient bones can crack open a narrative that's been accepted for decades and ask politely but firmly, what if we had it wrong? So that's what this is about. Who are the people buried in the Amazon? Where did that Pacific Signal AC truly come from? What does it mean for a genome to match across 10,000 meters of open ocean? And what were ancient humans genuinely capable of long before we gave them credit for any of it? We're going to work through the evidence carefully, not jumping to conclusions, but not flinching from the ones the data actually supports either. Before we get into any of that, I want to know where you are right now. What time is it? And where in the world are you listening from? Drop it in the comments. Someone's hearing this at sunrise, someone at midnight, someone in the middle of a Wednesday afternoon commute. That kind of thing is genuinely fascinating to me. Tell me. Now, let's start with what they actually found. The Amazon is not a friendly place for ancient bones. You have to understand what it means to try to preserve organic material in a tropical rainforest. The heat is relentless. The humidity never drops. Fungi, bacteria, insects, acidic soils, flooding cycles that repeat every year for thousands of years. Everything that biology and geology can throw at organic matter, the Amazon throws it. The same environment that produces extraordinary biodiversity actively destroys the evidence of the humans who lived there. In many parts of the basin, bones don't survive a few centuries, let alone a few millennia. So, when researchers find human skeletal material that is genuinely ancient, genuinely intact enough to work with, they're already looking at something rare, something the Earth almost swallowed entirely. The sites where these remains were found sit in a region where the forest floor holds layer after layer of human occupation. Archaeologists work through these layers carefully. The way you'd peel back pages of a document that was written, buried, written over, and buried again across thousands of years. The deeper you go, the older the material. But depth alone doesn't give you a date. What gives you a date is a combination of methods, and we'll get into the specifics of that in the next section. For now, understand that 7,000 years old is not a casual estimate. It's a number that comes from rigorous analysis of the material itself and the sediment surrounding it. It places these individuals in a time when agriculture was either brand new or hadn't arrived here yet, a time when the people who lived in this basin were doing so entirely on their own terms, entirely outside any contact with the old world, entirely outside anything that conventional history books would describe as civilization. What was actually recovered? Fragments. That's the honest answer. Not complete skeletons laid out in neat, anatomical order. Partial skulls, jaw sections, limb bones, in varying states of preservation. That's typical for this kind of site and this kind of environment. You work with what survived, and what survived even in fragments can tell you an enormous amount. Bone chemistry records, diet. The ratios of different isotopes locked in dental enamel tell researchers whether a person was eating primarily plants or protein, whether they grew up in this region or somewhere else entirely. Bone structure and density can indicate health, stress, physical labor patterns, disease. Even a fragment of FEMA carries biographical information if you know how to read it. These individuals, whoever they were, appear to have been living in the forest rather than passing through it. Their remains suggest long-term habitation. People embedded in a landscape, not visitors. That matters because it rules out the simplest dismissal of the find, which would be to say, these people that drifted in from somewhere exotic, died, and left no real trace on the local population. The evidence suggests otherwise. So why did this find stand out? Ancient South American remains are not unheard of. Sites across Brazil, Chile, and Colombia have produced human material going back thousands of years, but most of those remain, when analyzed genetically, fit a recognizable pattern. They connect to the populations we expect. The groups descended from the migrations that came down through North America, following the coastlines, spreading into the continent over tens of thousands of years. That's the baseline. That's what the data normally shows. These bones didn't show that. Or, more precisely, they showed that in something else, something additional, something that had no straightforward explanation within the standard model. That's where the questions start. And here's where the scientists themselves are careful, appropriately careful, to flag the complications. Incomplete skeletons mean incomplete information. Ancient DNA is fragile, and contamination is a real and serious risk. Modern DNA from researchers, from soil bacteria, from anyone who handled the material at any point and infiltrate a sample and corrupt the results. The best laboratories have protocols specifically designed to guard against this, but the risk never fully disappears. There are also disputes about the context of the find itself, about which stratigraphic layer the bones came from, about whether the dating is as solid as the headlines implied. None of those concerns erase the finding, but they're part of the honor story. And the honor story is what we're here for. So, how does a number like 7,000 years actually get attached to a piece of bone? It's a fair question, and the answer is more complicated than most headlines suggest. Dating ancient material isn't a single test you run and read like a thermometer. It's a process of building a case, cross-checking evidence, and ultimately arriving at a range you can defend rather than a single clean number you can carve in stone. Start with radiocarbon dating because that's the method most people have heard of and the one most relevant here. The basic principle is elegant. All living things absorb carbon from the environment. A tiny fraction of that carbon is a radioactive isotope called carbon-14. While an organism is alive, it keeps absorbing Bing carbon-14, maintaining a relatively constant ratio of that isotope to stable carbon. The moment it dies, the clock starts. Carbon-14 begins to decay at a known measurable rate, a half-life of roughly 5,730 years. Measure how much carbon-14 remains in a sample. Compare it to what should have been there at death, and you can work backward to estimate when that organism died. Simple, in principle. Genuinely complicated in practice. The first complication is calibration. The assumption behind basic radiocarbon dating is that atmospheric carbon-14 levels have been constant throughout history. They haven't. Solar activity, volcanic eruptions, ocean circulation patterns, and more recently industrial pollution have all shifted the ratio of carbon-14 in the atmosphere over time. If you don't correct for this, your dates can be off by decades or even centuries. Scientists address this through calibration curves painstakingly built from samples with known ages, tree rings that can be counted back thousands of years, coral records, ice cores. The raw radiocarbon measurement gets run through these curves to produce a calibrated date. What comes out the other side isn't a single year. It's a PR, probability range. Something like 95% confidence that this individual died between 5,200 and 5,400 years before present. That range isn't a failure of the method. It's an honest acknowledgement of its limits. Now, here's where it gets more complicated for skeletal materials specifically. You can date the bone itself, or you can date the material found around it, and those two approaches aren't interchangeable. Dating bone means extracting collagen, the protein matrix that survives inside bone long after the rest of the organic material degrades. Collagen can hold a radiocarbon signal for thousands of years under the right conditions. When the collagen is well preserved and uncontaminated, it gives you a direct date on the person. You're measuring the carbon from their own body, from what they ate and breathed during their lifetime. But collagen in a tropical environment is often compromised. Heat and moisture break it down. If the collagen is too degraded, the sample becomes unreliable and experienced laboratories will reject it rather than produce a number that looks precise but isn't. In cases where bone collagen can't be trusted, researchers turn to the surrounding context. Charcoal from a hearth near the burial, shells from a mitten associated with the same layer, seeds preserved in the same sediment. These aren't dating the person directly. They're dating the deposit, the layer of time that the burial appears to belong to. And that distinction matters enormously, because here's one of the oldest pitfalls in archaeological dating. The bones and the sediment aren't always from the same moment. Burial is a real phenomenon. Later communities sometimes dug into older deposits intentionally or accidentally. A burial from one era can end up physically surrounded by material from an entirely different era. The charcoal you date might be from a fire built centuries before or after the person was placed in the ground. Without careful stratigraphic analysis, careful reading of the layers and their integrity, you can end up with a date that technically measures something real but doesn't mean what you think it means. Disturbed stratigraphy is the nightmare scenario. Tree roots punch through layers. Burrowing animals mix sediments. Flooding redistributes material over and over across thousands of years in a basin like the Amazon. A researcher excavating a site has to reconstruct not just what's there, but whether it's still where it originally came from. Every layer is interrogated, the consistency of the soil matrix, whether artifacts or bones are oriented in ways that suggest natural deposition or disturbance, whether the sediment chemistry matches across horizontal distances. It's detective work applied to dirt. And then there's the reservoir effect, which is specifically relevant in Amazonian contexts. Aquatic systems, rivers, wetlands, and the organisms that live in them operate on a different carbon budget than terrestrial environments. Fish, freshwater shellfish, and aquatic plants absorb carbon that's already old, pulled up from deep water or sediment. A person who ate a lot of riverine food will have a radiocarbon signature that makes them appear older than they actually are. Their body chemistry reflects the old carbon in their diet. If researchers don't account for this, they can overestimate the age of skeletal remains from populations that lived close to rivers, which describes a significant portion of ancient Amazonian peoples. This is why no serious archaeologist publishes a date from a single test on a single sample and declares the question settled. What builds confidence is convergence. Multiple samples from the same context, direct dates on collagen, where preservation allows, cross-cheed against associated charcoal, against shell, against the broader stratigraphic sequence. When different materials from the same deposit produce dates that cluster in the same range, when the calibrated probabilities overlap, when the geological story told by the sediment matches the ages produced by the chemistry, that's when you can say something with real weight. The 7,000-year figure attached to these Amazonian bones isn't a single measurement. It's the product of this kind of accumulation, which is also why when scientists and critics push back on the dating, the response isn't to point to one number. It's to examine the entire chain of evidence and ask where specifically it might be weak. That's the standard. And the standard exists because getting the date wrong doesn't just produce an incorrect headline. It changes the entire story you're trying to tell about who these people were, when they lived, and what their existence means for our understanding of the ancient Americas. Dating bones is one thing. Reading the DNA inside them is another layer of difficulty entirely. When you imagine DNA, you might picture that famous double helix, a long, elegant ladder of information twisting off into the distance. In a living cell, that's not a bad mental image. The genome is made of stretches of DNA, millions of letters long, neatly packaged, constantly repaired. In a 7,000-year-old bone from a tropical forest, it's more like confetti. Heat, humidity, microbes, oxygen, and time all attack DNA. Enzymes chop it into shorter and shorter pieces. Chemical reactions alter individual bases. Strands break, ends fray. After a few thousand years, what's left is not a complete book of instructions. It's a pile of shredded pages with coffee spilled across them, some letters smudged beyond recognition. Ancient DNA laboratories are built around this hard reality. When researchers drill into a bone or tooth from the Amazon and try to coax out DNA, they're not expecting a pristine genome to come pouring out. If they get anything at all, they'll mostly get fragments only tens of bases long, little scraps. And those scraps are damaged in a predictable way. One of the classic signatures of ancient DNA is a particular chemical, L change. Cytosine, one of the 4 DNA letters, tends to turn into uracil over time, which shows up in sequencing as cytosine being read as thymine, especially at the ends of molecules. It's like finding that all the C's at the edges of your shredded documents have faded into T's. That damage pattern is annoying, but it's also gold, because modern contamination, the DNA from the scientist who handled the bone last week or from the person who dug it up 20 years ago, doesn't have that pattern. Modern DNA is long, mostly intact, and chemically fresh. Ancient DNA is short, ragged, and scarred in exactly the ways thousands of years of chemistry produce. This is the first rule of reading ancient DNA without fooling yourself. You don't just look at what letters you got. You look at how broken and damaged they are. If your supposed 7,000-year-old genome looks suspiciously healthy with long fragments and little damage, you have a problem. You're probably sequencing the living, not the dead. Which brings us to contamination, the eternal enemy of this field. If you were going to deliberately design a nightmare for ancient DNA research, you'd pick a small skeleton handled by dozens of people over decades, excavated from warm, wet soil, then stored in a museum drawer that opens in and out of room air. In other words, a very typical archaeological specimen. Every touch leaves skin cells, sweat, breath, microscopic droplets of saliva. Every one of those carries modern DNA. Modern DNA that is vastly more abundant and intact than the tiny fragile fragments still hiding inside the bone. So, the labs that do this work operate more like biohazard facilities than conventional genetics labs. Researchers suit up in full-body protective gear. Air is filtered and pressurized so that air only flows out of the clean rooms, never in surfaces. Tools and the bones themselves are treated with bleach and ultraviolet light to destroy stray DNA on the outside. The drill bits that pulverize bone are sterilized between samples. Even the reagents, the chemicals used to extract and amplify DNA, are routinely tested for background contamination. And still contamination happens. That's why the second rule is redundancy and skepticism. You don't just sequence once. You sample from different parts of the same bone or tooth. You run negative controls. Pretend samples, where you go through all the steps, with no bone material at all, to see if your lab is carrying stray DNA from one experiment to the next. You check whether the DNA you're seeing looks like it could belong to the archaeologists who excavated the site, or the lab staff, or common lab contaminants. When a team claims that an ancient Amazonian individual carries a genetic signal resembling a Pacific population, they're making that claim on the back of this whole apparatus. Clean rooms, damage patterns, controls, and repeated checks that the DNA really is old and really is from that bone. Once they're confident about that, then comes the question, okay, but what does it actually match? This is where population genetics steps in and where a lot of misunderstandings begin. A human genome is 3 billion base pairs long, but most of those letters are essentially the same in all of us. What population geneticists care about are the positions in the genome where humans differ. Places where maybe 40% of people have one letter, 60% have another. These variable sites are scattered across the genome like markers on a map. To compare an ancient genome to modern groups, scientists look at which variant is present at each of hundreds of thousands of these sites. Then they compare that pattern to a big reference panel. Genomes from living people and other ancient individuals from many regions, all catalogued and organized. The tools they use are statistical models that ask, given what we know about how populations split, mix, and drift over time, how likely is it that this ancient individual shares ancestors with population A versus population B? Models that look at shared chunks of DNA, shared rare variants, subtle correlations between markers, things no human FIRS could confidently detect by just glancing at sequences. So when you hear that an ancient Amazonian genome matches a population from the Pacific, it doesn't mean the scientists found a label in the DNA that says property of Polinia. It usually means something more technical and more modest. When you run this genome through various models, it shares more genetic variants, or more specific patterns of variation with certain specific groups than with other candidate groups in the database. It might mean that, in a who are you most similar to analysis, this Amazonian genome clusters slightly closer to some Pacific Islanders than to most other Native American groups, UPS. It might mean that particular stretches of their genome look like stretches found today in people from certain Pacific Islands and are rare elsewhere. That's a match in a statistical sense. It's not a one-to-one identity. It doesn't mean this person's parents got off a canoe from Tahiti. It doesn't even necessarily mean direct contact. It could point to deep shared ancestry in a population that no longer exists in pure form, or to a minor contribution from a distant group that was diluted over generations. And here's where the limitations start to bite. Any comparison is only as good as the reference it's compared to. We have a lot of genomes from some parts of the world. Europeans, East Asians, some Native American groups with long histories of contact with researchers. We have very few from others, especially small, remote, or historically marginalized communities, many of whom have understandable reasons to be cautious about genetic research. For the Amazon itself, and for many parts of the Pacific, modern sampling is still patchy. For ancient DNA, it's even worse. Whole regions have zero-sequenced ancient individuals. Others have a handful. Imagine trying to reconstruct the history of literature if the only surviving books from the 1800s were 3 novels from England, 1 from Russia, and a newspaper from Boston. Any new text you found would match one of those because that's all you have. So, when an Amazonian skeleton seems to show a genetic affinity to a particular Pacific group, one possibility is exactly what the headlines suggest, some kind of real biological connection. Another is that the true source population isn't in the database at all, and the models are grabbing the closest thing they can find. A ghost population from the ancient coastline of Asia, for example, might leave descendants, both in some Pacific Islanders and in early South Americans. If we've only sampled the islanders, that's who will light up in our analyses. There's another subtle trap: interpretation bias. Scientists are human. If you see a weak signal that aligns with a story you find exciting, early trans-Pacific contacts say, you might be tempted to lean into that narrative. Statistical tools can be pushed in different directions by seemingly reasonable choices: which populations you include or exclude, which time periods you focus on, which models of migration you assume are plausible. Change those assumptions and the Pacific signal might look sharper, or it might blur into background noise. This is why in good studies claims about matches are hedged with language like affinity, signal, component, and consistent with. It's not evasion. It's an attempt to be honest about what the methods can and can't tell you. So reading ancient DNA without fooling yourself means staying grounded in the chemistry. Are we really dealing with ancient molecules and humble about the statistics? Are we really seeing a robust pattern, or are we staring at a raw shack ink blot of noisy data and limited references? With that in mind, when we say these Amazonian bones matched a population from the Pacific, the real question becomes matched which Pacific people, in what way and against what background of missing information. To unpack that, we need to zoom out and look at who those Pacific comparison groups actually are and what stories their own DNA carries about movement across oceans and continents. When a paper says the Amazonian bones show an affinity to Pacific populations, it sounds specific. It sounds like we're talking about one island, one culture, one voyage across the ocean. We're not. In population genetics, Pacific is a convenient label for several overlapping things. Sometimes it means the people of remote Oceania, Polynesia, Micronesia, parts of Melanesia, whose ancestors mastered long-distance voyaging with outriggers and stars. Sometimes it includes near Oceania, places like New Guinea and the Bismarck Archipelago, where humans have been living for tens of thousands of years. And sometimes it's even broader: coastal East Asians, island Southeast Asians, indigenous groups from the rim of the Western Pacific. On a map, that's an almost absurdly vast zone, from Taiwan and the Philippines in the west, down through Indonesia, across to New Guinea and the Solomons, out into the scattered atolls of Micronesia, the high islands of Fiji and Samoa, then further still to Hawaii, Rapa Nui, and Aotea, New Zealand. In genetic datasets, any of these communities might be filed under the umbrella of Pacific, and each of them has its own deep, complicated history. Let's start near the western edge. New Guinea and nearby islands were settled very early, at least 40,000 to 50,000 years ago, by some of the same broad wave of modern humans that spread across Asia and into Australia. The people whose descendants live there today carry genetic lineages that split off from other non-African populations extremely early. In many analyses, Papuan and Australian Aboriginal group sit on long distinct branches of the human family tree, reflecting tens of millennia of relative isolation. Much later, another drama began to play out north and west of there, in what we now call island Southeast Asia and coastal China and Taiwan. Sometime around 5,000 to 3,000 years ago, farming populations speaking Aranesian languages started to expand out of Taiwan and the surrounding mainland. They brought with them crops, domesticated animals, pottery styles, and seafaring technologies that allowed them to hop from island to island, the Philippines, then Indonesia, then further east into the Pacific. When these Aranesian-speaking farmers reached places like New Guinea and the Bismarck archipelago, they didn't sail into an empty ocean. They met and mixed with the descendants of those very ancient populations who had been there since the Pleistocene. Genetically, culturally, linguistically, a fusion began. The people we call Pacific Islanders today, especially those in Polynesia and parts of Micronesia, are the descendants of multiple such fusions. Analyses of modern Polynesian genomes, for example, show a blend of ancestry related to East Asian and island Southeast Asian farmers, plus ancestry related to Papuan-like groups from near Oceania. The exact proportions vary from island to island, and sometimes even village to village, shaped by which group stopped where, when, and how many times they intermarried. So when a genetic study labels a reference group Polynesian or Papuan or Aranesian, those names are shorthand. They're not single, pure bloodlines. They're statistical summaries of many individuals, each of whom already carries the results of multiple migrations, contacts, and internal population splits, stretching back thousands of years, and there's more layering. Over the last few centuries, colonialism, slavery, trade, and labor migration brought Europeans, Chinese, South Asians, and others into Pacific Islands in large numbers. Many living Pacific communities are now genetically mixed with these later arrivals too, though careful studies try to account for that by focusing on participants with deep local ancestry or by using methods that will isolate the older components of the genome. All of this means that when an Amazonian genome shows a specific affinity, we're not matching it to some untouched time capsule population from 4,000 years ago. We're matching it to modern people whose genes have been shuffled and recombined through wave after wave of human movement. That's not a flaw in the science. It's the human condition. Every population on Earth is a palimpsest. Older stories written over, but not entirely erased by newer ones. But it does force some humility about what the match can mean. Imagine you're trying to reconstruct an ancient language from a handful of surviving phrases. You compare them to modern languages. You have recordings of English, Spanish, Mandarin, Murray. Your mystery phrases sound a bit more like Maui than the others. Does that mean the speakers came from New Zealand 3,000 years ago? Not necessarily. It might mean both your lost language and Maui descended from some older tongue, no longer spoken, and Maui just happens to be the closest surviving cousin. Genetic signals work the same way. The Pacific component that shows up in Amazonian DNA might reflect direct contact with the ancestors of a species, a Pacific modern island community, but it could just as plausibly reflect shared ancestry from a population that once lived somewhere along the vast Pacific Rim, coastal East Asia, island Southeast Asia, even part of the now submerged Sunda or Sahul shelves, whose descendants later contributed to both early South Americans and to some of today's Pacific Islanders. In technical terms, It's a shared ancestry signal, not a stamped passport. The tools population geneticists use, things like f-statistics, admixture graphs, and clustering algorithms, are quite good at saying, this ancient Amazonian person shares a bit more genetic drift with modern group X than with group Y. They're much less precise at saying, this means their grandmother was from island Z. Complicating it further, the signal in the Amazon is weak. We're talking about a small fraction of their ancestry, often modeled as only a few percent, nudging them slightly towards certain Pacific or Australasian-related groups compared to other Native Americans. That's tiny compared to, say, the clear 50-50 mixing you might see in someone with one parent from Europe and one from East Asia. It's the kind of subtle tilt that could be asked by just a few additional generations of mixing, or that might only show up clearly with very sensitive statistics. So why does that matter? Why does a few percent detectable only with math, most of us don't do in our heads, trigger so much discussion? Because of where it is. The Amazon is not a Pacific island. It's not even a coastal region facing the Pacific. It's deep inside South America, past the Andes, in one of the most inland forest-choked basins on the planet. In most simple models of how the Americas were people, you have an initial migration from Northeast Asia across Bingia into the far north, then a spread southward through the continents. All of that movement is supposed to be on the American side of the Pacific, with no need to invoke back and forth contact across the ocean. In that tidy picture, Amazonian groups should look like variations on a broadly similar Native American genetic theme, shaped by local drift and later events, but not carrying exotic signals from half a world away. So when buried inside that expected pattern, you find even a faint echo that seems to point back across the Pacific, an excess of shared variance with Papuan or Polynesian or certain indigenous Philippine groups compared to what your models predict. It rings alarm bells. Maybe the models are missing a piece of deep pre-Berian structure in Asia. Maybe a now vanished coastal population hugging shorelines from southern China to the Americas left crumbs of ancestry on both sides of the ocean. Maybe there were later, more direct contacts across the Pacific much earlier than the Polynesian voyages we know about from archaeology and oral history. Or maybe it's all an artifact, a quirk of sampling, a ghost of some unsampled Siberian group that just happens to look a bit Pacific in our limited databases. The weak nature of the signal means all of those possibilities stay on the table. It doesn't let us jump straight to canoes in the Amazon 7,000 years ago, but it also doesn't allow us to ignore it. Because if it turns out to be real, if that little tilt toward the Pacific is the footprint of an actual population-level event, it would force us to redraw the clean lines in our map of how people reached and moved through the Americas. To understand why, and to see what exactly it would be challenging, we need to look at that standard story. The one map you've probably seen in textbooks that seems to explain everything with a single arrow from Siberia to the tip of South America. You've probably seen some version of it. A globe, or maybe a flattened map of the northern hemisphere with a thick red arrow starting somewhere around Siberia, curving across what is now the Bering Strait, then fanning out down through North and South America. Sometimes there are a couple of dates scribbled along the way. 20,000 years ago up north, 13,000 further south, 11,000 near the tip of Patagonia. One grand migration, one direction, one story. It's a powerful image because it's so simple. It implies that peopling the Americas was basically a one-step process. A group walks across a land bridge from Asia, then gradually spreads out like water pouring downhill. Reality is messier, but that map isn't entirely wrong. It's just compressed. It folds an enormous amount of complexity into a single arrow. Here's the cautious version. Most researchers agree that the ultimate source of indigenous American ancestry lies in Northeast Asia. Sometime during the last ice age, people related to ancient Siberian and East Asian groups moved into a region that no longer exists on modern Mars. P.S. Bingia. Bingia wasn't just a narrow bridge you sprinted across in a hurry. Sea levels were roughly 100 meters lower than today, and the continental shelves between Siberia and Alaska were exposed. That created a land mass as big as some European countries combined. It had rivers, hills, wetlands, steppe, maybe patches of scrubby forest. For thousands of years, it was a habitable world in its own right. Genetic data suggests that the ancestors of most Native Americans didn't just cross Bingia like a highway. They likely spent a long time there, isolated from their Asian relatives to the west and from the Americas proper to the east. This Bingian standile could have lasted several thousand years, perhaps beginning 25,000 or more years ago. While massive ice sheets locked down much of northern North America, eventually the ice shifted as the climate warmed toward the end of the Pleistocene. Glaciers retreated. Two big ice sheets that had walled off the interior of the continent began to pull back, creating possible passages south. At the same time, coastlines changed. Kelp forests, rich marine ecosystems, and narrow ice-free refuse may have lined the Pacific edge. And this is where the state NDERD story branches into two main routes. In one version, people move south through an inland ice-free corridor east of the Rockies between receding continental ice sheets. For a long time, this was the textbook answer. You can imagine bands of hunter-gatherers following herds through a deglaciated valley, gradually filtering into the Great Plains. In another version, people come down a Pacific coastal route instead, traveling along shorelines by foot and possibly small boats, exploiting marine resources, hopping between refuges of land not buried under ice. This coastal highway wouldn't have been a nice clear trail. Parts of it are now underwater, and the evidence is frustratingly sparse, but many archaeologists think it may have opened earlier than the interior corridor, making it a plausible path for the first southerly expansion. At this point, the clean map arrow starts to wobble because timing matters. Some of the oldest, widely accepted archaeological sites south of the big northern ice sheets are already present in the interior of North America by around 14,000 to 15,000 years ago, maybe a bit earlier, depending on which dates you trust. There are sites in Chile and South America that some researchers argue are at least 14,500 to 15,000 years old, possibly older. If those early southern sites really are as ancient as their proponents claim, then people had to move from Bingia to the tip of South America in just a couple thousand years. That's a staggering rate of spread. We're talking on the order of a few hundred kilometers per century, generation after generation across unfamiliar landscapes full of large animals, new climates, new diseases. It's not impossible. Humans are very good at expanding into empty or sparsely occupied territory. Small founder groups can move fast, especially along coasts and river systems where resources are predictable, but it leaves little room for the leisurely trickle implied by that gentle map arrow. This is why timelines are still hotly debated. Push a site's date back by even 1,000 years, and you're rewriting how quickly people must have moved, which routes they used, and how long they had to diversify into the many distinct cultures we find in the archaeological record. When you look specifically at South America, the picture gets even busier. Genetic studies of indigenous groups suggest that the continent wasn't populated by a single uniform wave that fanned out smoothly. Instead, you see evidence for multiple pulses, splits, and bottlenecks. Different branches of that original bringing derived population appear to have moved south at slightly different times or along slightly different paths, then gotten somewhat stuck in their own regions, accumulating their own distinct histories. The Andes, for example, create an enormous ecological and cultural barrier running north-south, populations on the Pacific side, in the highland valleys, and on the Amazonian side of the mountains faced very different environments and over time develop different patterns of movement and interaction. Some models propose an early pulse that hugs the Pacific coast and then spreads inward in certain places, and a somewhat distinct branch that enters more directly into eastern South America. Others see signatures of later waves of movement overprinting older patterns, groups expanding out of the Andes onto the lands or moving back northward from southern refugees as climates shifted. Whatever the exact configuration, one consistent theme emerges. South America is not a single genetic block. It's a mosaic of lineages that have been diverging, mixing, and rhizolating for at least 12,000 to 13,000 years, maybe longer. Now place the Amazon into that mosaic. On a map, the Amazon basin looks like a giant green smear. But in lived reality, it's a vast, intricate space. It's bigger than many continents if you include all its tributaries and surrounding forests. Distances that look trivial on a classroom globe translate into weeks or months of travel on foot. At the same time, it's crisscrossed by what are essentially natural highways, rivers. The Amazon River and its tributaries form one of the most extensive freshwater networks on Earth. For people who know how to navigate them, these waterways link distant regions in ways that dense forest on foot simply does not. A village hundreds of kilometers away, as the crow flies, might be effectively next door if it lies along the same river corridor, and almost unreachable if it sits behind several ridges and swamps off that route. So, the Amazon is both connected and fragmented. On the one hand, river travel allows trade, marriage exchange, and the flow of ideas over giant distances. On the other hand, the forest itself, the seasonal floods, the patchwork of soil types, and the sheer scale of the basin can isolate groups for long stretches of time. A small community tucked up a minor tributary might go generations with minimal contact beyond its immediate neighbors, especially if political tensions or resource competition make long-distance journeys risky. Genetically, that tends to produce pockets of deep differentiation. Groups separated by only a few days paddling can end up surprisingly distinct if they've been drifting apart in relative isolation for thousands of years. Add in occasional pulses of newcomers pushing into the basin from the Andes, from the Ghana Shield to the north, or from the Atlantic-facing coast to the east, and you get a complex overlay of ancestries. In the standard one-arrow map, the Amazon is usually just another part of the spread south phase. People arrive, fill the forest, end of story. But zoom in, and it becomes clear that the Amazon is more like a labyrinthine crossroad. Water highways linking far-flung regions, ecological barriers that slow or divert movement, and countless opportunities for small founding groups to branch off and follow their own trajectory. Now, layer the Pacific question on top of that. If all you had was the simple arrow from Siberia down the spine of the Americas, you wouldn't expect any Amazonian group to carry a genetic whisper that points back toward populations thousands of kilometers across the ocean. The standard framework allows for multiple waves from Northeast Asia, some early coastal movement, some inland variation, lots of local drift, but all of it is supposed to happen on this side of the Pacific. That's why the Pacific leaning signal in those 7,000-year-old Amazonian bones matters so much. It doesn't just add a detail, it nudges against the basic architecture of that map. The timing of the Beringian standstill, the number and nature of migration pulses into South America, the way the Amazon's river networks connected or insulated early populations. To see just how much room there is within that standard story for something unexpected to hide, and how the Amazon could act as both a filter and a funnel for strange ancestries, we need to look more closely at the basin itself as a genetic crossroads. Picture a canoe slipping into the current just after dawn. Mist hangs over the water. The forest on either side feels impenetrable, a wall of green. From the river, though, the world opens up. The current grabs the hull, and suddenly you're moving faster than you could ever walk, carried along a liquid highway that existed long before roads and will outlast all of them. This is how a lot of the Amazon works. If you imagine the forest as a giant trackless jungle, it looks like the perfect recipe for isolation. It's hot, wet, full of insects, and dense understory. You can't see far. Trails overgrow on foot. Progress is slow and punishing. But flip your perspective. Stand in a dugout canoe instead of on the forest floor, and the map changes. The Amazon basin is threaded with more than 100,000 kilometers of navigable rivers and streams. The main Amazon itself is a freshwater superhighway, but it's only the trunk of an enormous branching system. Hundreds of tributaries feed into it. Those are fed by smaller blackwater and whitewater streams, seasonal channels that swell and shrink, streams of water that appear only during high flood. To people who know how to read these waters, the forest is not a barrier. It is organized by flow. You can start in a small creek in what is now Peru, paddle downstream into a modest tributary, merge into a larger river, and without ever stepping onto land, eventually reach the Atlantic Ocean thousands of kilometers away. Along the way, you pass places where other rivers come in from the Andes. Others arrive from the Ghanas. Some angle up toward what's now Venezuela, others down toward what's now Bolivia or central Brazil. Each confluence is not just a geographical node. It's a social one, a place where people from different stretches of the basin can meet, trade, intermarry, exchange stories, exchange genes. In that sense, rivers don't just move logs and fish and sediments. They move DNA now at where people like to live. If you're trying to make a living in the Amazon as a forager or a farmer, you don't just randomly plop down in the forest. You look for specific things: access to fish, fresh water, game trails, good soil. The floodplains that line the big rivers, those broad flat areas that go underwater for part of the year, are prime real estate. When the water rises, it brings nutrients. When it falls, it leaves behind rich silt. Fish crowd into floodplain lakes. Fruits ripen along the margins. It's no accident that many modern and historical indigenous settlements cluster along these margins. Buried in those floodplains is something else. Terra pra, dark earth, is a human-made soil found in patches across the full charcoal, broken pottery, food waste, and other organic material. It doesn't occur naturally in those forms and concentrations. It's the byproduct of long-term habitation, centuries of people living in one place, burning, cooking, composting, discarding, carefully or accidentally engineering of the most fertile soils in the region. When archaeologists map Terrapa, they're not just mapping dirt, they're mapping neighborhoods and towns that existed hundreds, sometimes thousands of years ago. Some of these patches are huge, on the order of many hectares, suggesting sizable, stable communities. So now combine that with the river network. You have long-lived settlements sitting on or near the river highways. People stay in one area for generations, building up dark earth, raising crops, domesticating plants, managing forests. At the same time, the river in front of their village brings visitors and carries their children and grandchildren away to marry into other groups upstream or downstream. Genetically, that's an engine for both stability and mixing. A small group can split off, travel a few days by canoe, find a new bend in the river with slightly higher ground and decent fishing, and start a new village. That's a classic founder event. A handful of people representing only a slice of the genetic diversity of their home community. At first, that new village is almost a genetic copy of the parent group, just smaller. But over generations, a few things happen. Random chance. E shuffles genes around differently. Maybe they take spouses mostly from one direction on the river, and not the other. Maybe a new family arrives from a completely different tributary with a slightly different ancestry. Maybe a disease wipes out a subset of lineages. Give that process 1,000 years or more, and the new group can end up carrying a noticeably different genetic signature, even though culturally they might still share language, myths, or technology with their neighbors. Now zoom out and imagine not just one such split, but hundreds happening over and over again along thousands of kilometers of river. Some offshoots fizzle and disappear. Others grow large, generate their own spin-offs, and become regional hubs. Some receive repeated pulses of people moving in from the Andes, from the Atlantic coast, from upland plateaus. Others sit tucked so far up minor streams that they see outsiders only rarely. That's one way you get unusual patterns in Amazonian DNA. Not because there was a single dramatic invasion or mass replacement, but because rivers allow small groups to move far and fast, then settle in ways that amplify chance events. A tiny founding group carrying a slightly atypical ancestry can, under the right conditions, become the ancestors of a whole region. The tricky part is the physical evidence of all this isn't easy to find. In a desert, stone walls and hearths can sit exposed for thousands of years. In temperate grasslands, buried sites survive in relatively stable soils. In the tropics, the environment is actively trying to erase the past. Heat, humidity, and acidic soils break down bone and organic material quickly. Roots push through ancient deposits. Seasonal floods scour away layers or bury them under meters of new sediment. Wooden structures, baskets, textiles, and even many stone features vanish into the churn of the forest floor. That means the Amazon is probably full of places where people lived, died, and were buried for centuries. But the archaeological traces are faint or gone altogether. The rare sites that do preserve bone or DNA, like the ones that produce those 7,000-year-old remains, are exceptions, not the rule. So, our picture is automatically skewed. We see spectacular mounds, geoglyphs, earthworks, and terrapita where conditions happen to favor preservation or later detection. We miss countless small camps, temporary clear, ings, earlier occupations that might tell us when certain ancestries first arrived or how widely they spread. On a genetic map, this can look like sudden appearances and disappearances. One group shows a surprising ancestry. Their neighbors don't, and with no dense archaeological record between them, it's tempting to draw straight lines. These people must have come directly from over here. But in a landscape where evidence is patchy and perishable, there could have been entire chains of intermediary communities now totally invisible to us. This is where contact networks come back into the story. The Amazon doesn't exist in isolation from the rest of South America. Those same rivers that flow eastward from the Andes carve deep valleys that connect the highlands to the lowlands. People on the Pacific side can move inland along river systems, cross passes, then drop down into tributaries that feed the Amazon. Groups in the foothills might move seasonally between upland and land zones, acting as brokers of both goods and genes. To the east, smaller rivers and portage routes link the Amazon to the Atlantic coast. To the north and south, waters interlock with the Orinoco, the Paran, and other major basins. In some seasons, you can literally drag a canoe a short distance over a divide and find yourself in a different drainage entirely with access to a different set of communities. Archaeological finds, ceramics with shared styles, similar technologies, crops that clearly originated in one region but show up hundreds of kilometers away suggest these connections were real and active. Salt, pigments, stone, shells, ritual items, and stories moved along these paths. So did marriage partners, war captives, refugees, and ambitious leaders looking for new territory. Put all this together, and the Amazon stops looking like a remote, isolated backwater and more like a slowly shifting web that links coast, Andes, and interior. A person born near the Pacific coast could within a generation or two have descendants living in a foothill. Their grandchildren might marry into a land group along an Andean tributary. Their great-grandchildren could be living deep in the central Amazon, fishing a blackwater river under trees that their coastal ancestors never saw. Genetically, the signal from that original coastal population doesn't broadcast evenly across the continent. It's through D is it way along specific routes, amplified in some pockets, diluted or erased in others. When you finally stumble on a burial in just the right soil, in just the right microclimate, and sequence the DNA, you're catching a tiny glimpse of that tangled history. So, when those ancient Amazonian bones whisper of a connection to people across the Pacific, the question isn't just how did a strange signal get into the forest, it's how did the forest and its rivers move that signal around, and where else might it be hiding invisible to us for now? To even start answering that, we have to think about mechanisms. What kinds of events, migrations, or long-distance exchanges could plant a Pacific-leaning ancestry in one corner of South America and let it percolate through this watery maze? In other words, if the Amazon is a crossroads, what paths lead into it from the Pacific side? Let's start with the boldest version of the story. Imagine people with an ancestry related to Pacific-facing groups showing up very early, not as visitors skimming the coastline, but as part of the original human wave into the Americas. In this first scenario, you don't tack a Pacific signal onto the end of the narrative. You bake it in from the beginning. Maybe somewhere in Northeast Asia, there wasn't one homogeneous source population that crossed into the Americas, but several related groups. Some leaned genetically closer to the ancestors of present-day East Asians. Others carried affinities to the ancestors of later Pacific Islanders or Australapan groups. They all inhabited overlapping landscapes, intermarrying, splitting, rejoining. Then, during one narrow window, when ice sheets and sea levels allowed, multiple branches crossed into the Americas in a staggered sequence. Some of those branches would go on to dominate the genetic landscape of both continents. Others would remain small, tucked away in corners, swamped by later expansions, or simply unlucky in the demographic lottery. If the people buried in that Amazonian floodplain belonged to one of these earlier, more Pacific-leaning branches, the pattern in the DNA makes a particular kind of sense. In the data, you'd expect their genome to show a deep connection to other indigenous Americans because they are fundamentally part of that same founding story, yet carry a subtle, consistent tilt towards certain Pacific or Australasian populations, ions that isn't as strong in later or neighboring groups. Crucially, the timing would match. If this was baked into the founding population, you'd see the signal in very old remains elsewhere too, especially in other early sites in South America and maybe faintly in North America. As you move forward in time, the signal would either persist at low levels in multiple lineages or become concentrated in pockets that happen to preserve it by chance. Geographically, it wouldn't appear as one isolated dot. It would look more like a faint watermark visible in early layers across a broad area, even if later demographic waves blur it out. Scientists test this kind of scenario by comparing genomes from different time slices. They use statistics that ask questions like, is this Amazonian individual more closely related to Group A or Group B after accounting for shared ancestry? And does the pattern change if we bring in even older skeletons from other regions, if ancient samples from, say, the Andes or the southern cone of South America, especially those older than or similar in age to the Amazonian ones, show the same kind of Pacific-leaning tilt that supports an early continent-wide contribution from a related source population? If the signal only appears in one small pocket and only at certain times, then this deep baked-in scenario starts to look less likely. Now, contrast that with a second possibility. The Pacific signal doesn't arrive with the first Americans. It shows up later, traveling in pulses along the edges of the continent and filtering inward. In this version, picture a chain of coastal societies strung along the Pacific margin of South America. People are harvesting shellfish, fishing, perhaps managing coastal resources in ways we're only beginning to appreciate. Some of these communities preserve a bit more ancestry from a Pacific-facing population, whether because of their original founding or because they maintain contact along the coast. Over centuries, that ancestry moves inland, not as one heroic voyage across the entire continent, but as a relay. A coastal group trades with people in a low coastal valley. Those valley communities have ties to higher elevation Andean groups. The Andean foothill people have seasonal camps in the upper reaches of Amazon tributaries. Each link doesn't feel like an epic migration. It feels like what we talked about every Arlia, people visiting in-laws, merchants carrying salt or obsidian, refugees moving a few valleys over to escape conflict. If this were the mechanism, the genetic predictions look different. You'd expect the earliest Amazonian samples to lack the specific Pacific-leaning signal. It would only appear after a certain date, whenever those contact networks crystallize strongly enough for genes to ride, along with goods and stories. Spatially, it might show up first along certain corridors, valleys that connect the Pacific to the Andes, rivers that make it easy to drop from the highlands into the forest. Sites positioned near those choke points would be most likely to show the mix signature. Farther away, deep in the lowlands, the signal might be weaker, more diluted, or arrive later. In the genomes, you might see something like a layered cake, the deeper ancestry shared with all indigenous Americans, plus an extra component that looks closer to particular Pacific Rim populations than you'd expect from a simple one-time founding event. That extra component might be restricted to certain regions or time periods rather than spread evenly. Researchers probe this by building models. What if, with take a baseline Amazonian ancestry and then mix in X% from a coastal-like population at 3,000 years ago? Does that recreate the patterns we see? They compare the fits of these models to alternatives, see which ones require fewer awkward assumptions, and test whether the same explanation works across multiple individuals and sites. If you can't find that intermediate ancestry in coastal or highland remains from the right periods, this later contact model starts to wobble. But if ancient DNA from Pacific valleys and Andean foothills shows a stepwise gradient, strongest near the coast, weaker but still detectable in the highlands, and faint but present in Amazonian groups, that's exactly the fingerprint you'd expect from genes riding along well-traveled routes. There's a third scenario that's subtler, but important to keep on the table. Maybe neither group ever actually visited the other. Maybe the resemblance between Amazonian and certain Pacific populations is a ghost of a shared past, not evidence of a direct bridge. In this version, wind the clock back even farther to a population somewhere in Asia or perhaps along the ancient coastlines now drowned by higher seas. From this broad nasal pool, different branches split off. One branch gives rise to the people who eventually moved down the Pacific Rim into island Southeast Asia near Oceania, maybe later into remote Oceania. Another branch gives rise to a subset of the people who cross into the Americas. Both inherit some distinctive genetic quirks from their common ancestors, then go their own ways for tens of thousands of years. By the time we meet them in the genetic record, they've been separate for so long that they're unquestionably distinct lineages. But statistical tools are sensitive enough to pick up that they share a few biases that other groups don't. When you compare them to a wide panel, they look slightly more like each other than you'd expect if their only relationship was distant cousins through a big Pan-Asian population. That can easily masquerade as evidence of direct contact, when in fact it's just both parties remembering in their DNA an ancestor that lived long before either the Amazon or Polynesian canoes existed. In terms of predictions, this shared source scenario implies that the signal should be relatively ancient and broad. You wouldn't expect a sharp on-off switch in time in the Americas because it wasn't introduced by a specific migration event into South America. It came in with the original founding populations, just in low, diffuse amounts. You'd also expect that if you sample widely across Asia, especially ancient remains, you'll find populations that sit in between genetically speaking and make the resemblance between Amazonian and Pacific groups less mysterious. Once those intermediates are in the analysis, you might see the apparent direct connection weaken or disappear. Geneticists test this by adding more potential source populations into their comparisons, especially as new ancient genomes from Siberia, China, and Southeast Asia become available. Often, what looks like a direct affinity in a simple model turns into a more complex but less dramatic story in a richer model. Multiple splits, minor admixture events, extinct lineages leaving faint shadows. Across all three scenarios, one thing stays constant: uncertainty. The same pattern in a handful of genomes can sometimes be explained by very different historical stories. With limited samples from the Amazon, fragmentary remains from the Pacific and enormous gaps in Asia's ancient DNA are echoed. It's easy to overinterpret or to see your favorite hypothesis in the noise. So, scientists pit these scenarios against each other. They simulate data under different migration histories and see which ones best match the real genomes. They look for consistency. Does the model that explained the Amazonian signal also work for other sites? Or does it only fit if you fine-tune it for one location? They wait for new discoveries, another skeleton in a different floodplain, a burial in a high Andean valley, a coastal midden with preserved bones, to either reinforce or unravel what looked like a neat solution. Right now, all three paths remain open. An early Pacific-leaning founding branch, a later infusion through coastal and riverine contact networks, a deep shared ancestor that makes populations on opposite sides of the ocean look oddly familiar. To decide which of these is actually written into the bones of the Americas, we have to ask a harder question that goes beyond statistics and family trees. Given the technology and seafaring skills people had thousands of years ago, could they really have crossed or closely followed the Pacific early enough to matter? To even ask that question, Tion, you have to step out of the lab and onto the water. Picture a shoreline 10 or 15,000 years ago. No concrete harbor, no steel hulls, no GPS, just a strip of sand, a line of surf, and a horizon that might as well be the edge of the world. People are already masters of this space in ways we sometimes underestimate. Long before anyone thinks about crossing an entire ocean, humans are doing short sea hops. They're paddling across estuaries, crossing narrow straits, hugging shorelines in small craft that leave almost no archaeological trace. A log hollowed by fire and stone tools, a bundle of reeds lashed together, a frame of driftwood covered in animal skins. All of these can carry a family. Their tools may be a dog from one side of a bay to the other. We know this not because we've found those specific boats. They rot, they sink, they vanish, but because people show up on islands you simply can't walk to. By at least 40,000 to 50,000 years ago, humans are in Australia and New Guinea. To get there from mainland Asia, even when sea levels were lower, you still needed boats. Not necessarily sleek canoes slicing through the open ocean, but some kind of watercraft capable of repeated crow singing, of handling currents and winds that don't always cooperate. Later, we see people reaching islands in the Mediterranean in Japan, along the coasts of Southeast Asia. Archaeologists find stone tools, hearths, butchered bones on islands that were isolated even at the lowest sea levels. The verdict is clear. Humans have been comfortable with water crossings much longer than we once assumed. So in a basic sense, yes, people could cross bits of the Pacific early on, if by cross you mean short stretches between visible pieces of land. But that's not what most people imagine when they hear Pacific contact. They picture a canoe vanishing over the horizon, weeks at sea, guided only by stars and swells, heading for a continent the navigators have never seen. That's a different beast entirely. The Pacific is huge. Not big lake huge, planet-sized, huge. The Polynesian voyages that eventually crisscrossed it were among the most extraordinary feats of navigation in human history. And they were recent in archaeological terms, mostly within the last 3,000 to 3,500 years. By then, Pacific navigators had developed highly specialized skills, reading swells, memorizing star paths, recognizing UD shapes that form over islands, feeling tiny changes in wind and current. They had robust canoes, sophisticated knowledge of where islands actually are, and deep oral traditions about routes and landfalls. When we ask whether people could have crossed the Pacific early enough to influence founding populations in the Americas, we're really asking, did anything like that exist much earlier in a less documented form? That's where the evidence gets thin and careful language becomes important. For a full deliberate trans-Pacific crossing deep in prehistory, you'd want several kinds of support, not just a genetic hint in one or two skeletons. You'd want boats, or at least strong indirect evidence of them. Actual preserved hulls are rare before a few thousand years ago because wood and plant fibers decay, but sometimes you get impressions of planks, carvings that depict boats, or toolkits that only make sense in a maritime context. Specialized fishhooks, harpoon heads, boat-building adzes. You'd want coastal sites that speak the language of seafaring, shell mittens layered over centuries, camps perched at natural harbors, work areas full of fish bones from species you can only catch off shore, storage pits for dried fish and sea mammals. The more intensively people are living from the sea, the more plausible long-distance voyaging becomes. You'd want stepping stones, a trail of island sites or coastal stopovers that show a pattern of movement. Not just one lonely site on a distant shore, but a gradient, earlier dates on islands closer to the homeland, later dates farther away, similar tool styles or dietary signatures that look like a trail of breadcrumbs scattered across the ocean. And you'd want that same gradient in the genetics. If people are really leaping across parts of the Pacific and leaving a lasting mark, the ancestry shouldn't appear like a single sharp spike in an inland floodplain. It should ramp up as you move toward the presumed route, stronger on certain islands, moderate on the facing coasts, faint but detectable farther inland. Without some combination of those elements, interpreting a faint genetic resemblance as evidence for early heroic ocean crossings becomes risky. That's why archaeologists talk about smoking guns, a chicken bone on a South American beach that turns out by its DNA to be from a Polynesian lineage, a crop like sweet potato, native to the Americas showing up in Polynesia centuries before Europeans. These are the kinds of finds that light up the debate because they imply not just driftwood and accidental flotsam, but deliberate or repeated contact. Even then, nothing is straightforward. That chicken bone might be intrusive, mixed in from a later layer. The sweet potato might have floated, its seeds carried by birds or currents, rather than in the belly of a canoe. Every apparent smoking gun becomes a target itself, retested, redated, reinterpreted. The archaeological record along ancient coasts makes things even harder. During the last ice age, when many of the relevant migrations were happening, sea levels were much lower. The prime real estate for early coastal peoples, the sheltered bays, river mouths, gentle beaches are now underwater. If there were camps, boat sheds, fish processing areas along those shores, a lot of them are drowned, buried beneath meters of sediment on continental shelves. So, we're in an awkward position. The kinds of sites that would most clearly document early intensive seafaring are precisely the ones least accessible to us now. Underwater archaeology is possible, but it's too good to be true. Nickily challenging, expensive, and we've only scratched the surface. That's why extraordinary routes demand extraordinary evidence. It's not that long-distance Pacific crossings thousands of years ago are impossible. Humans are clever, adaptable, and have surprised archaeologists more than once. It's that, the more dramatic the proposed voyage, thousands of kilometers of open ocean, no intermediate landfalls, no prior track record of comparable journeys, the more lines of independent evidence we should expect before rewriting foundational chapters of human history. Contrast that with more modest scenarios, short sea crossings along coasts, island hopping where you can always see the next island, or at least know it's there from stories a few days away. Those are much easier to reconcile with the patchy evidence we have. A community that fishes from dugout canoes traveling 10 or 20 meters offshore is already within reach of nearby islands on a clear day. A small shift in wind, a storm, a misjudged current, and you can end up somewhere new. Sometimes you make it back and add that island to your mental map. Sometimes you don't. In that kind of world, genes, ideas, and objects can creep together. N significant distances along shorelines without anyone ever setting out to cross the Pacific. Each step of the journey is mundane, a trip to a neighboring bay, a seasonal move to a better fishing ground. It's only in hindsight, when you add up 1,000 small hops, that it looks like a grand migration. Open ocean crossings, by contrast, require you to deliberately let the coastline disappear, to trust knowledge systems that tell you there's land far beyond the horizon. For the early periods relevant to the first settlement of the Americas, we don't yet see clear, uncontested evidence of that level of maritime technology in the regions that would matter. So, when a single Amazonian skeleton whispers about a connection to distant Pacific populations, scientists have to weigh that whisper against what we know and what we don't about early seafaring. Could an early branch of Pacific-leaning people have followed the North Pacific coast into the Americas using relatively simple boats, staying close to land the whole way? That's within the realm of possibility, and it's a scenario many researchers take seriously. Could someone have launched from Polinia or island Southeast Asia and sailed directly to South America 7,000 or 10,000 years ago with no trail of intermediate traces? That's a much taller order. Until we find the right combination of coastal sites, maritime tools, island chains, and genetic gradients, the safe answer isn't a confident yes or a dismissive no. It's a kind of disciplined humility. People were capable of more on the water than we once thought. But if we're going to claim they spanned the largest ocean on Earth early enough to reshape the story of the Americas, we need more than suggestive patterns in a few genomes. We need anchors in the ground. And that brings us to the tug-of-war between the genetic hints and the archaeological record. What the dirt, bones, and artifacts are actually saying about contact and exchange across the Pacific. When archaeologists talk about contact, they're not just thinking about people. They're thinking about stuff. Stone tools, pots, ornaments, animal bones, seeds, material things that move when people move, or trade or copy what their neighbors are doing. In many ways, objects are the vocabulary of ancient contact. They're how we eavesdrop on conversations between groups that vanished thousands of years ago. One of the most basic clues is tool style. Imagine two communities living hundreds of miles apart, separated by dense forest and unfamiliar rivers. For generations, each group has its own way of shaping stone, different preferences for raw material, different ways of flaking a cutting edge, different ideas about what a proper spear point or knife should look like. Then, at some point in the archaeological record, you see something odd. In one region, a new kind of tool appears, a chipped stone blade with a very particular shape, or a polished axe with a distinctive cross-section. You dig farther, and you realize this style already existed somewhere else, earlier in a different valley on a different river system. Something connected those places. It doesn't automatically mean one group packed up and migrated wholesale to the other region. It could mean a few families moved and brought their toolkit with them. It could also mean occasional visits, gift exchanges, or even just someone seeing a stranger's tool, liking it, and copying it back home. Ceramics tell even richer stories. Clay remembers hands. The way a pot is formed, the mixture of clay and temper, the thickness of the walls, the finish, ring temperature, the painted patterns. These are all cultural signatures. When distinctive ceramic traditions jump suddenly across distances, archaeologists start wondering whether pots moved, potters moved, or potting ideas moved. In some parts of the world, like the Mediterranean or the Near East, you can actually trace detailed trade networks this way. Amber used for wine and oil appear in harbors far from where they were made. Pottery styles ripple outward like rings in a pond. In the Amazon, the picture is murkier, but not empty. For a long time, the stereotype was that Amazonian people were small, scattered bands, leaving almost no durable trace. That image is crumbling. Archaeologists are finding elaborate ceramic traditions in many parts of the basin. Painted pots, inside designs, even fine wares associated with particular regions and time periods. In some places, you see pottery traditions that look remarkably similar across hundreds of kilometers of river. Similar shapes, similar motifs, similar ways of building and firing the clay. That doesn't happen by accident. Rivers were highways and pots were passports. Ornaments and prestige goods can be even more direct. A bead, made from a shell that only lives on a distant coast. A piece of obsidian traced chemically to a volcano hundreds of miles away. A jaguar tooth pendant showing up in a region where jaguars are rare or absent. These are like neon signs, flashing. Something traveled along the Pacific. And in parts of the Americas, we see this kind of long-distance movement. Clearly, obsidian from specific Mexican sources turns up far to the south. Spondylus shells from warm tropical waters appear in highland and Indian tombs. In the Amazon, objects carved from exotic hardwoods, feathers from distant bird species, and carefully ground stone axes hint at exchange networks spanning major rivers and ecological zones. Then there are the living artifacts, plants, and animals. Domesticated species are some of the strongest indicators of sustained contact because they don't usually move themselves in stable, cultivated populations. People carry seeds, cuttings, tubers. They transport live animals. They transplant entire ecologies in miniature. When a crop native to one continent suddenly appears on another, that's a big red flag for human-mediated contact. The classic example that fuels Pacific America debates is the sweet potato. It's native to the Americas, but it shows up in Polynesia before Europeans arrive, and with names in Polynesian languages that resemble some American terms. That looks suspiciously like trans-Pacific transfer. On the flip side, chickens may have traveled in the opposite direction. Some controversial finds of ancient chicken bones in Chile got people arguing about pre-Columbian Polynesian poultry. The DNA evidence there has been challenged and reanalyzed multiple times. But the very fact that a chicken bone can ignite that kind of debate tells you how powerful plant and animal transfers are as markers of contact. Inside the Amazon itself, you see something similar on a more local scale. Crops like manio, cacao, and certain varieties of peppers don't just sit neatly in one little patch of forest. They appear and reappear along distant river systems, often associated with similar kinds of soil modification or settlement patterns. You get terra pria, the famous Amazonian dark earth, enriched by human activity, showing up like islands of fertility along major rivers. Landscapes that look engineered rather than just those engineered landscapes, plus the distribution of crops south, will suggest that ideas and plants were moving along these water highways for thousands of years. That's a form of contact and exchange you can trace without ever invoking ocean crossings. But here's the key thing genetics adds. You can move a crop or a tool-making style with almost no movement of genes. Trade is lopsided that way. A canoe full of pottery, seeds, and ornaments can sail upriver, offload its cargo, and return home without leaving a lasting genetic signature. Maybe one marriage alliance here and there, a visiting artisan, a captured slave. But in population-level terms, a trickle of people can carry a flood of ideas and objects. Genetically, that looks like continuity. Archaeologically, it looks like change. That's why tying artifacts directly to migrations is so tricky. Suppose you find a distinctive ceramic style in two far-flung regions and you date them carefully. The earlier one is clearly older. It's tempting to draw an arrow on a map and say, migration from here to there. And sometimes that's justified, especially if it lines up with changes in settlement size, diet, and burial practices. But there are always alternative scenarios. Maybe just a few expert potters moved, married into a local community, and taught their techniques. Maybe the style spread along a trade route as a fashionable aesthetic. Maybe different groups independently converged on similar solutions to the same practical problem: how to make a pot that won't crack in a certain kind of fire. In the Pacific America's context, this is exactly why the debate is so persistent. Take the sweet potato. Does it represent Polynesians reaching the Americas, Americans reaching Polynesia, or some now lost intermediate population shuttling back and forth? Were there multiple transfers over centuries, or one lucky voyage that launched a crop on a new continent? The same kind of argument plays out with those controversial chicken bones, with certain fishhook styles, with canoe depictions in rock art, with obsidian flows that seem to leap further than expected. In almost every case, some researchers see human voyages and deliberate contact, while others see more mundane explanations: misdated layers, later contamination, convergent designs, ocean drift. The Amazon adds another complication. The record there is uneven. Huge areas of forest have never been systematically surveyed. Many sites we do know about were discovered accidentally by road builders, by farmers, by satellite imagery, hinting at geometric earthworks beneath the canopy. Where archaeologists have been able to dig properly, the surprises keep coming. Enormous pre-Columbian earthworks, causeways and canals, artificial lakes and fish ponds, mounds and plazas that imply large organized communities, not scattered bands. We're only beginning to see how dense and connected some Amazonian societies might have been. Ceramic traditions that once looked isolated now seem to form broader patterns along river corridors. Botanical remains, charcoal layers, and animal bones are filling in stories of managed forests, fishes, and long-term landscape engineering. But it's still patchy. For every well-studied site, there are hundreds of kilometers of forest with no data at all. The most ancient layers are often the hardest to reach: deep, disturbed, or eroded. Floodplains shift, rivers migrate, entire villages might have been swept away 1,000 years ago. So when a genetic study claims a deep connection between an Amazonian skeleton and a faraway Pacific population, archaeologists turn to their toolbox: pottery, stone, bones, seeds, earthworks, animal remains. Do we see anything in the ground that demands an explanation involving trans-Pacific contact? Do we see foreign materials coming in? Exotic styles appearing suddenly? Crops that shouldn't be here yet? So far, the answer is tentative. We see rich, complex Amazonian systems of contact and exchange, powerful enough to move ideas, goods, and perhaps small numbers of people over immense distances inside South America. We see coastal and riverine networks along the Pacific and Atlantic fringes, capable of carrying prestige goods and crops far from their points of origin. What we don't yet see is a clear, unbroken archaeological trail that runs from a Pacific homeland across thousands of kilometers into the heart of the Amazon in the timeframes implied by the most striking genetic signals. That doesn't mean it never existed. It means for now, the material record is whispering about local and regional connections much louder than it's whispering about trans-oceanic ones. And that tension between what the dirt is saying and what the DNA is hinting at is exactly where alternative explanations and scientific pushback start to emerge. When a result feels similar to a plot twist, scientists don't just lean back and enjoy the story. They start poking holes in the script. Those 7,000-year-old Amazonian bones with a genetic signal that looks eerily like a population from across the Pacific. To a cautious researcher, that's not just fascinating, it's suspicious. Not because it can't be true in principle, but because it's exactly the kind of surprise that statistical methods are very good at faking when the conditions are just right or just wrong. The first thing skeptics reach for is sample size. Ancient DNA studies almost never have the luxury of huge data sets. You're dealing with a few individuals, sometimes just one or two, with patchy genomes and lots of missing data. That's like trying to infer a whole book from scraps of sentences. When you compare a tiny, noisy genome against a big library of modern and ancient populations, strange coincidences are guaranteed to happen. A handful of shared mutations here, a cluster of similarities there, and suddenly the software says, "Hey, this Amazonian person looks closer to this specific group than to their neighbors." The math behind those comparisons assumes certain things, like the errors being random or the missing data not having a pattern. In reality, ancient DNA is messy in very non-random ways. Damage tends to hit certain bases. Coverage is better in some regions of the genome than others. Some stretches are just gone. With small sample sizes, those quirks can get amplified. Imagine flipping a coin only 5 times and getting 4 heads. That doesn't mean the coin is rigged. It just means you didn't flip enough times. In genetic terms, one skeleton with limited usable DNA is like 5 coin flips. A critic looks at that and says, "Okay, interesting hint. But how many individuals did you test? How many independent lines of evidence do we have? And when you tweak the settings, change which parts of the genome you analyze or how you handle missing pieces, does that Pacific-like signal stay rock solid, or does it wobble and fade?" That leads straight into the second big issue, reference bias. Any statement like this, ancient Amazonian person is closest to population X hides a crucial detail closest among which options. Geneticists don't compare an unknown sample to every human who ever lived. They compare it to a panel of living and ancient genomes that happen to have N sequenced and curated. That reference panel is never complete, and it's never neutral. If your library of comparison populations includes lots of data from, say, Polynesians, some Southeast Asians, and a smattering of Native American groups, but very few from certain Amazonian regions, or none from long-vanished groups in northern South America, you're stacking the deck. The algorithm is forced to say, "Among the things you gave me, this is the best match." It's not allowed to say, "Actually, what I really want is a population you don't have in your database." In practice, this can create illusions. An ancient genome might share a suite of variants with some modern Pacific group, not because they had direct contact, but because both descend from a deeper, older population that isn't represented in the modern samples. If you don't have that ancestral group in your reference panel, the model grabs the next best stand-in. It shrugs and says, "Pacific, it is." Skeptics are very aware of this. They'll ask questions like, "Did you include enough diverse Native American populations? What about groups from Central America, the Caribbean, or coastal South America that might have vanished or been heavily admixed after 1492? Did you test alternative reference panels to see how stable the Pacific connection really is?" Then, there's a subtler problem: convergent signals and deep shared ancestry. Genetic similarity doesn't always mean recent shared history. Sometimes it points to chapters that go way back toward the front of the human story, long before the peopling of the Americas or the settlement of the remote Pacific. Think of humanity's expansion across Asia as a long branching river. One branch eventually leads into Bingia and the Americas. Another, much later, flows out into the islands of the Pacific. But both branches may carry traces of an older pattern that formed upstream. Subtle frequencies of certain variants, shared bits of ancestry from a long-gone population that once lived somewhere in Eastern Asia or near the Pacific Rim. Over tens of thousands of years, drift, selection, and random chance will alter those patterns, but not completely. Two groups separated by oceans and millennia can still carry echo traces of that ancient source. If you then compare them without a good map of all the intermediate populations, those shared echoes can look like direct contact. That's convergent signal in genetics. Not convergent evolution in the sense of dolphins and ichthyosaurs evolving similar shapes, but convergent patterns of ancestry that can mislead if you interpret them in too narrow a frame. So when a paper says this Amazonian individual shares a distinctive signal with population Y in the Pacific, the cautious translation might be this Amazonian individual and population Y both preserve a rare ancestry component that our current models don't fully capture. That's still exciting. It suggests a more tangled, multi-branched human history than the simplest textbooks show. But it doesn't automatically draw a line of canoes between Polynesia and the Amazon 7,000 years ago. Because of all this, peer review in ancient DNA is obsessed with methods. Before anyone argues about grand narratives, reviewers want to know how clean is this data. Was the excavation careful? Were the bones handled with gloves stored properly, shielded from modern DNA? Did the lab include damage patterns that are typical of ancient DNA, like characteristic chemical changes at the ends of fragments, as a sanity check that the sequences really are old? They'll dive into contamination, ination estimates, how much modern human DNA is mixed into the sample. Does the mitochondrial DNA show multiple individuals? Do the sex chromosomes make sense for the skeleton's anatomy? Then they go after the models. Which statistical tools did you use? Statistics, admixture graphs, principal component analysis, approximate Bayesian computation. What assumptions do those models make about population splits, migration rates, and random drift? How sensitive are your conclusions to those assumptions? Did you try simpler explanations first, like deep shared Asian ancestry, before reaching for dramatic ones like trans-Pacific contact? Or did you jump straight to the most newsworthy scenario? This is why critiques of splashy findings often sound harsh. It's not personal, it's triage. Reviewers know how easy it is to tell a beautiful wrong story with the right-looking graphs. So, what would it take for skeptics to start shifting toward acceptance of a strong Pacific Amazon link that early? First, replication. One skeleton with a Pacificel-like signal could be a fluke, an outlier, or even a lab error. But if you start seeing the same pattern in multiple individuals from different sites dated independently, the noise explanation gets weaker. Imagine 5, 10, 20 ancient Amazonian genomes spread across centuries and river systems, all showing the same weird connection in slightly different ways. At some point, the probability that it's all an elaborate statistical mirage drops. Second, robustness across methods. Critics would want to see the signal holding up under different analytical approaches and different reference panels. Run the data with and without certain modern populations. Add more ancient Asian genomes as they become available. Test more complex demographic models. If the Pacificite component stubbornly persists no matter how you torture the data, that's a good sign. It's not just an artifact. Third, temporal and geographic logic. Does the timing of the signal make sense relative to what we know about Pacific population history? Are you claiming a link to a group that didn't even exist yet, 7,000 years ago in its current form? Or does the connection line up with plausible ancestral populations in East or Southeast Asia, whose descendants later spread into the Pacific Islands? Does the signal cluster in certain Amazonian regions may be closer to the Atlantic or to Andean corridor? Or is it scattered randomly? Patterns in space and time matter. They can distinguish a one-off contact from a deep, shared prehistory. Fourth, convergence with archaeology and other lines of evidence. Skeptics aren't waiting for a Polynesian canoe preserved in Amazonian mud, but they would be more comfortable if other data started pointing in the same direction. Unusual tool traditions, odd plant distributions, marine species inland, or coastal sites with mixed cultural signatures, even isotopic analyses, chemical fingerprints in bones that hint at marine diets versus terrestrial ones could contribute. If individuals with that Pacificel-like genetic signal also show life histories that don't quite fit local expectations, people will pay attention. Finally, transparency and humility in the storytelling. Researchers who say, "Here's the pattern we see, here are 3 possible explanations, and here's what we'd need to distinguish them," tend to earn more trust than those who declare, "We found proof of X," in a field this uncertain, with data this fragile. Critics want to see that the authors understand the difference between a tempting narrative and a demonstrated conclusion. For now, the Amazon-Pacific connection in these 7,000-year-old bones sits in that liminal zone, too intriguing to ignore, too shaky to canonize. The real test of the idea won't come from a single dramatic paper, but from what happens next, what gets dug up, sequenced, and argued over in the coming years. And that raises the obvious question, what exactly would have to turn up to push this from strange hint to solid chapter in the human story? If you want to turn a strange hint into a solid chapter, you don't start with a single spectacular skull. You start with a map. Not a map of migration arrows and bold claims, but a quiet, unglamorous checklist of places where we still have almost no ancient DNA at all. On that list, the Amazon is practically a blank. Think about that for a second. One of the largest river systems on Earth, home to some of the deepest linguistic diversity in the Americas, a region that has clearly been inhabited for thousands of years. And in terms of ancient genomes, it's a handful of individuals scattered across a vast, complex landscape. If you want to know whether a Pacificite ancestry really threaded itself into this region, you need more than an odd signal from one site. You need dozens of individuals from up and down the river system, different tributaries, floodplains and uplands, ancient villages on bluffs above the water, and temporary camps deep in the forest. And you can't stop at the forest. If there was any movement that could plausibly connect Pacific populations to the interior of South America, the Andes and the Pacific coast are the obvious corridors. Mountain passes, coastal valleys, river mouths that open onto the sea. Right now, these areas are also severely undersampled in time and space. A few high-profile mummies, some well-preserved skeletons from dry caves, a small number of coastal sites, enough to sketch early branches of Andean history, not enough to map subtle threads of ancestry pushing east, west, or along the shore. Then there are the islands. If you're contemplating any scenario that even flirts with ocean crossings, you have to look hard at island chains and coastal stepping stones. Islands off Ecuador and Peru, the Galapagos, remote Pacific archipelagos, whose settlement histories are still being refined with every new dig. Ancient genomes from those places, especially very early ones, could show whether traces of Amazon-like ancestry ever appear on the Pacific side, mirroring the Pacific traces seen in the Amazon. That back-and-forth signal would be a very different story from a one-way mysterious genetic echo sitting only in the interior. But collecting ancient DNA from all these regions is not just a question of drilling more bones. It's about how you do it. For this particular debate, more genomes is not enough. You need well-dated, well-contextualized genomes. A skeleton pulled from a looted burial with a vague label, like late prehistoric, is almost useless for this question. No matter how good the DNA looks, you don't know exactly how old it is. You don't know what layer it came from. You don't know what materials were around it, what the burial practices were, what the environment looked like at that time. Any surprisingly specific signal from a sample like that is just a genetic Rorschach test. People can see whatever migration story they already believe in. By contrast, imagine a small cluster of burials on a clearly stratified terrace above an Amazonian river. The lowest layer is radiocarbon dated, cross-G'd with multiple samples from charcoal and bone anchored to a firm W. Endow in time, say 7,200 to 6,800 years ago. The sediment has been studied, whether it was floodplain, forest, or open scrub when those people lived there. There are tools, pottery, or lack of pottery, plant and animal remains, maybe even evidence of hearths and post holes. Now, the DNA comes from individuals whose lives you can place in a tight geographic and cultural frame. If they show an unusual ancestry component, you can relate it to specific ways of living in a specific environment at a specific moment in history. That turns a weird signal into a testable pattern. The same logic applies in the Andes and along the Pacific coast. Bones from a rock shelter that's been disturbed by centuries of reuse, erosion, and burrowing animals tell a much noisier story than burials from a sealed cemetery with clear stratigraphy and multiple independent dates. This is why serious ancient DNA papers now devote enormous space to context and chronology. The genetics might grab the headlines, but the dates and dirt are what make the conclusions believable, and genetics is only one language that the past speaks. To really test ideas about long-distance connections, you need other complete military evidence. One powerful tool is isotopes, the tiny chemical signatures locked into teeth and bones that record what people ate and sometimes where they grew up. The ratio of certain elements like strontium, oxygen, and carbon can vary from place to place depending on geology, rainfall, and diet. A person who spent childhood on a salty marine-rich coast will often carry a different isotopic pattern from someone who grew up eating mostly inland plants and river fish. So imagine this, a set of ancient Amazonian individuals whose DNA carries that Pacific-like signal and their isotopic profiles show spent near the sea, not deep in the forest. That would raise eyebrows. Or the reverse. A group on the Pacific coast whose genomes look ordinary for the region, but whose isotopes suggest they were born far inland in a basin that looks chemically more like the Amazon. That would suggest mobility, people moving across ecological zones, potentially carrying their genes and stories with them. Then there's language. Linguistics can't reach back 7,000 years with the clarity of a genome or a radiocarbon date. Sound shifts and word meanings drift. Whole languages disappear or blend. But U-news, Yule patterns, shared vocabulary that resists easy borrowing explanations. Strange structural similarities between distant families can hint at ancient contacts or shared roots. If linguists find deep, consistent quirks, tying certain Amazonian language families to those spoken by coastal or island groups linked genetically to the Pacific, that won't prove ocean voyages. But it will add another small weight to one side of the scale. Archaeology adds its own clues, shared tool types that appear abruptly far from their expected range, plant and animal species that turn up in places they shouldn't be without human help, distinctive pottery styles that seem to jump coastlines or mountain chains instead of diffusing slowly through intermediate regions. Each of these alone is ambiguous. Together, if they all begin pointing in the same broad direction, they can transform a controversial genetic signal into a coherent multi-line story of movement, contact, or shared ancestry. Layered on top of all this is a moving target, the methods themselves. The way scientists recover DNA from bones today is not what it was 10 or even 5 years ago. New extraction techniques are pulling usable genetic matter, IAL, from warmer, wetter climates that were once thought hopeless. Specialized sampling of the petrous bone in the skull or even dental calculus, the hardened plaque on teeth, has dramatically boosted yields from degraded remains. Improved lab protocols mean lower contamination. Better sequencing technology means more complete coverage from tiny fragments. And on the computational side, demographic models are growing more realistic. They can explicitly incorporate pulses of migration, population bottlenecks, ghost lineages that leave echoes in multiple descendants. All of that matters because when the models change, the conclusions can flip. A dataset that in 2015 looked like clean evidence for a direct connection might in 2026 be better explained as the fingerprint of Dar-e, unsampled ancestral group, once you add more reference genomes from East Asia or the Americas and run more flexible models. Conversely, a faint, statistically fragile signal in early analyses might become much clearer with higher coverage genomes and newer tools that handle missing data more intelligently. What looked like noise can condense into an unmistakable pattern when you upgrade both the data and the mat. This is why no one discovery gets the final word. The next well-preserved skeleton from a remote Andean valley. The next coastal site that yields human teeth alongside unusual tools. The next batch of isotopic readings from Amazonian burials. Any one of these could push the story in a new direction. They might strengthen the case for an early unexpected link between Pacific populations and the Amazon. They might refine it, shifting the connection back in time to a deeper Asian ancestor, or forward in time to a more recent coastal contact. Or they might undercut it entirely, showing that what looked like a transoceanic thread was really an artifact of missing data and optimistic modeling. The point is not that we can't know, it's that what we know today sits on a scaffolding that's still under construction. And if a few puzzling bones from the Amazon can force us to redraw parts of the human story once, they can do it again. So what happens to our picture of the past if that Pacific-like signal turns out to be real and not just a statistical mirage? What else in the standard story starts to wobble when you tug on that thread? If that thread holds when we tug on it, the first thing that changes is not a headline like Pacific Voyagers discovered the Amazon. What changes is our sense of simplicity. For a long time, the story of the Americas has been drawn with one thick arrow. People walk or boat along the Bering Land Bridge, fan out across the continents, diversify locally, and that's basically it until relatively recent contacts. The details get filled in routes, timings, bottlenecks, but the backbone is a single sweeping movement. These bones in the Amazon and the Pacific, like signal in their DNA, are a reminder that history rarely cooperates with single route stories. The responsible takeaway right now is this: the deep history of South America is probably more layered, more braided than the clean map suggests. Multiple movements, some big, some tiny. Groups splitting and reconnecting, filtering through different landscapes, sometimes leaving strong genetic footprints, sometimes only the faintest trace. Here's what's actually solid so far. We know these individuals are old. The dating, as far as we can tell, is careful. We know their genomes are mostly what you'd expect for early Amazonians. And we know that, when you compare their DNA to a big panel of global populations, there's a statistically detectable pull towards certain Pacific groups that isn't obvious in everyone else. That's the rock. What's still sand is the interpretation. We don't yet know whether that signal reflects a real later contact between people already living in South America and some Pacific-connected group, or a much older shared ancestor back in Asia, whose genetic echo shows up both in the Pacific and in these Amazonian individuals, or some combination filtered through complex population history we still haven't sampled. We don't know if it represents a small, short-lived thread, maybe a few dozen people moving through, or a larger, more sustained connection. And we definitely don't have enough data yet to claim trans-Pacific voyages 7,000 years ago as a settled fact. That uncertainty isn't a weakness. It's a clue to how much of this past is still invisible. The Amazon doesn't preserve bones the way a dry cave in the desert does. Tropical heat, humidity, acidic soils. These are not friendly to collagen and DNA. For most of the history of archaeology, that meant the rainforest's earlier chapters were practically silent. Only in the last decade or two have new Ds made it possible to coax genetic and chemical whispers out of remains that would have been written off as hopeless. We're suddenly hearing voices from times and places that used to be blank spaces on the chart. When you start turning up those voices, your mental map has to change. Maybe the Amazon was not a remote, isolated backwater that people drifted into once and then stayed put. Maybe it was a crossroads where different waves, different stories intersected. Some came down river corridors from the Andes. Some filtered in from the Ghanas or the Atlantic coast. Some may carry echoes of older journeys that once linked shores we wouldn't normally connect on a standard classroom map. That's the deeper shift. This mystery hints at a past where humans are not just spreading out in one direction, but weaving back and forth across enormous spaces, experimenting with rivers, coasts, and maybe, just maybe, open ocean more boldly and earlier than we've been comfortable assuming. I'm curious where you land on this. Do you lean toward an ancient Asian ancestor that happens to show up in both Pacific and Amazonian groups, a later coastal connection that sent genes inland along the rivers? Do you think we're underestimating early seafaring and should take trans-Pacific experiments seriously? Tell me your theory in the comments, and just as importantly, what kind of evidence would actually change your mind? Is it a cluster of well-dated burials on both sides of the Andes sharing that same Pacificel-like signal? Matching isotopes that show coastal childhoods in inland graves, shared artifacts or crops that clearly jumped routes they shouldn't have? I'd love to hear how you're thinking about it and where you're listening from. The story of human movement looks very different if you're watching this from Lima, or Manaus, or Hawaii, or Berlin. If you found this journey through ancient DNA and rainforest history thought-provoking, take a moment to like the video. It really does help more people find it. Subscribe if you'd like to follow along as new discoveries force us to redraw the map again and again. And drop a comment with your thoughts, your questions, and your location. After all, the places you live, the routes your ancestors took to get there, are part of this story